Okay, so as most of you, I'm at my home office operating in Warsaw at the Center of New Technologies. And it's been a very nice event so far. So I'm glad, Marek, that you decided to have this conference in the end. Since we come from multiple backgrounds, I will try to keep my talk as simple as possible, and we're going back to molecular biology, molecular biophysics. I will show you what techniques my group uses to determine the functional dynamics of biomolecules using HPC resources, because this dynamics is typically important for function. So what we do, we simulate biopolymers, not only proteins. We heard a talk about proteins. From proteins, mainly enzymes, but we also simulate RNA or DNA molecules. What is important that these kinds of biopolymers, they do not work in isolation. But oftentimes they associate into dimers or trimers or large macromolecular complexes. So, for example, the ribosome, the protein production machine in the cell, is a complex of over 50 proteins and long non-coding RNAs. And another example, a viral capsid, is also a collection of many proteins. For the simplest, for even the simplest plant viruses. So the basis of our work are atomistically detailed structures obtained from experimental methods that are listed in this slide. So why we are interested in these macromolecular complexes? So one of the reasons is that these are drug targets. For example, the ribosome is a target for many antibiotics. Small molecules, such as shown here, an example of an aminoglycoside antibiotic, can turn on and off this large macromolecular complex. So these are the systems. What we have to remember is that these molecules undergo thermal motions. I'm not sure if you see the movie, but believe me, it jiggles and wiggles. And I'm showing an example of a small protein simulated via molecular dynamics in explicit solvent. And the movie might look quite messy, but the message here is that everything fluctuates, undergoes thermal motions, and these motions are important for function. If you strip the simulation movie of water molecules, it reveals some collective motions of protein fragments. So you can further on analyze them and detect what is interesting and what is important. You can quantify these motions, and the timescales of these motions are often spanning many scales. So from atomistic fluctuations on a femtosecond scales to what we heard today, polypeptide folding up to millisecond. And the systems that we simulate are, at least in my group, are up to the size of about 300 angstroms. So these we have to account in our sort of detection of molecular motions to be able to be predictive. We have to account for these different time scales. Now, to know how molecular conformations evolve in time, we need to determine energy, potential energy of each conformation to be able to characterize it and evolve it. And we use a simple molecular mechanics model of biomolecules following the Born-Oppenheimer approximation. And we assume that atoms are point charges, point interacting centers that interact via Hookean harmonic springs. We also account for electrostatics and the volume of each atom. And based on this simple model, we define the energy function. And going from the picture to the analytical formula, this is how it looks. We have the function is a sum of two body terms for certain atom types. And what is important is that we have interaction parameters for different atom types. And these parameters are typically based on spectroscopic, experimental, and quantum mechanical methods calculated for small building blocks. And we take them for all atomistic simulations. We take them from the databases. Now, once we have the energy function, so we can calculate energy for each conformation, we want to evolve the system. And there are many methods. I will stick to molecular dynamics, but there is also normal mode analysis or, for example, Monte Carlo simulations. So we evolved the system following the Newton's equations of motion in small time steps. And the algorithm, an example of an algorithm is one of the common ones like velocity Verlet. And we generate with the time step of, let's say, 1 femtosecond or 2 femtoseconds, millions of conformations. The bottleneck are typically non-bounded interactions, but we have some ways to calculate them, for example, using periodic boundary conditions, and for electrostatics, particle mesh evolt methods. Now, the first ND simulation of a protein was performed many years ago in 1977. And this was for a small bovine pancreatic trypsin inhibitor protein of 58 amino acids. The simulation took about 50 picosecond overall. And in 2013, Martin Karplus, I don't know if you can see the pointer, but not he and others, and also for other things, he received the Nobel Prize for these kinds of simulation techniques. Now, what we can do now for small proteins in explicit solvent, we can simulate microseconds routinely. How big a systems we can simulate? So a couple of years ago, two groups of Michael Feig and Yuji Sugita, they performed a simulation of a bacterial cytoplasm, which was about 100 million atoms. Shorter timescale, only 20 nanoseconds, but the box dimensions were quite large. So indeed, we can go to large systems. However, then we have to balance and have a smaller time scale, which means, going back to the slide with the time scales, that we have to pick the system size depending on the biological problem we want to address. We also have to bear in mind that there are some drawbacks of the force fields, so these parameter sets, that they were generated for small building blocks, and for some larger macromolecular complexes and their interactions as a fragment of bacterial cytoplasm, their interactions become sometimes too attractive. So we need to also develop force fields for these kinds of simulations. And also the MD method and the integration algorithms are bound with errors because they are mostly based on the Taylor expansion. So my group, we are end users of HPC resources. We currently use NAMD. We run on CPU clusters, but also a bit these ND techniques can be solved on GPU architectures as well. And depending on the system size, we go up to 500,000 atoms. We pick certain number of nodes for our applications. There are also specially dedicated machines for MD, like the Anton machine of the show company. And I saw tomorrow there will be a talk from Professor Taiji from RIKEN. He will also speak about such dedicated machines that he has developed. So the purpose of MD is generate conformations, and from which we can calculate time averages according to the ergodic hypothesis. So extensive sampling is a must. And standard MD is sometimes not sufficient because conformations get trapped in local minima. So there are many methods that enhance this sampling, and I would like to focus on one of which is quite intuitive. We do not have to represent a molecule in all atomistic description, but we can represent it in a reduced representation, so-called coarse grain. Let me see the time. Okay. So coarse graining, what does it mean? We reduce the representation and we gather a certain number of atoms into one pseudo-atom or effective particle. And for us, a very common mapping is of one nucleotide represented as one bead, or one amino acid represented as one pseudo-atom. So this way, we gain a lot. There is a payoff because, for example, if we take the system in explicit solvent of 200,000 atoms and we coarse grain it with one bead mapping, we lower the number of atoms, the degrees of freedom, by 3 orders of magnitude. However, there is of course a price to pay. We can simulate this system, this with a smaller representation for a longer time scale, but the price is that typically the force fields, the parameter sets, and the formulas to calculate the energies are for all atomistic descriptions because they are based on some experimental data and QM calculations, so they are easily accessible. But if we want to map our own system in a coarse grain representation, we have to find a formula and a set of parameters to evolve the system in a physically reasonable way. So we have, a couple of years ago, we have developed a tool that should aid and help in automatic parameterization of force fields. And I will not speak about this tool, but just to mention, it is downloadable from our website. But this optimization is made more automatic by using heuristic methods such as evolutionary algorithm or particle swarm techniques. So if you'd like to test it for your coarse grain representation, you are welcome to do so. Now, I would like to, after this introduction about MD simulations, I would like to go to the examples, showing you why the internal dynamics is important to consider in general. So the first example is of an enzyme, protease, of the HIV-1 virus that I simulated a couple of years ago, probably a decade ago. And this is an enzyme that is important for the HIV virus replication in general. And this is also a drug target. So on the left-hand side of the slide, you see examples of approved drugs that target HIV protease. These approved drugs compete with the substrate, and the substrate is shown in green in the active site, in its binding site. The enzyme is quite small, but what is important is that on the top, so covering the substrate, what you see are the flaps, so-called flaps, and these flaps are quite mobile. So many years ago, some NMR data suggested that these flaps move on a sub-nanosecond or nanosecond scale, and they were believed to be important for substrate association and binding, because in order for the reaction to take place, and this reaction is the cleavage of that substrate, the substrate is the peptide, the two molecules must associate and then form a binding complex. So we have designed a coarse-grained model to study the dynamics of that enzyme, and I will show you a movie if you see it. So this is the substrate that associates with the protease, but what it waits for the opening of the flaps. So from the top you see the flaps, and they are really quite mobile. The movement is around 30 angstrom. So it waits for the flap opening and then binds. Flaps close over. Now, this is an artificial cleavage, and then the products may dissociate. It's a coarse-grained model, so that's why I'm saying that the cleavages are artificial. So in a way, in a similar manner, competitive inhibitors, so the drugs that compete with the substrate, must associate with HIV protease in a similar manner. In case you haven't seen the movie, these are stages of the association of the ligand with HIV protease, showing how the flaps widely open. Now, the important thing is that the viral genome mutates of quite fast, and so the resulting proteins, including HIV protease, have in the end different sequence, often also different structure. So some of these mutants become drug resistant, and the question is why? Is it only because of the different interactions in the binding site, or different dynamics? Because some of these mutations are not in proximity to the active site. They are not local, they are positioned further away. So what we were investigating some time ago is, do these mutations affect flap dynamics? And it turned out, you see on the right-hand side, there are examples of how flaps open in the wild-type enzyme and in one of the mutants. And what you see is the flap tip distance. So the flap opening frequency as a function of the simulation time here in microseconds, because it's a coarse-grained model. So as a function of time in the wild-type enzyme, you see that the frequency of the opening, of flap opening, is quite large. But for one exemplary mutant, you can see that the flap dynamics is suppressed. And this might have important implications for the functioning of that enzyme, because the flaps become more rigid. And also the key would be, for example, can we design inhibitors that would enter the active site without flap opening? And indeed, this was the case. Some of these inhibitors do so. But as I spoke in the beginning, what is important is also the environment. So most of these simulations, we have to consider water molecules, ions, other macromoleculars, because cells are crowded. So in principle, there is in the system, we do not have only one substrate and one enzyme, but we have many of these. And apart from water molecules, we have also other crowding agents. Now, and I will show an example how crowding, how these macromolecules that are present in the cell can affect enzyme dynamics. And I will show it for a different enzyme, not HIV protease, but the enzyme protease of another virus, which is hepatitis C virus. So it's also a small protease that also cleaves peptide substrates, and it's essential for HCV multiplication, and also this enzyme is also a target for drugs, anti-HCV drugs, and one of these drugs is telaprevir. Now, we have been studying this enzyme, and one of the interesting things is that it has a cofactor, which is shown here in violet on the crystal structure, and which is quite long, extended, and necessary for the enzyme function. However, in the crystal structure in X-ray crystallography, it wasn't resolved because it's quite flexible. So Natalia, who is responsible for that project, she built a model of this cofactor, and you see how long these tails are, believed to be disordered, on the right-hand side. And what we did is we thought that these tails must be flexible, but what is their function? It's not 100% known, at least for all the parts. So we were interested in how the function of this enzyme is affected by macromolecular crowders. And here is one story that comes with some experiments. In our lab, what we did, this was done by Agnieszka. She studied the reaction of that enzyme with some peptide substrate in dilute buffer conditions, and also in the presence of synthetic crowders, polyethylene glycol molecules that are often used in laboratory settings to study crowding. Just to mention that the crowded conditions in experiments haven't been used for long. I think this is only in the last decade that people started to acknowledge that it is important to study reactions in some cellular mimicking conditions. So what she found is that the initial or maximal velocities of the reaction depend on the conditions, and the velocities are slowed down in the presence of polyethylene glycol molecules, and this is what you see in the left graph. So let's say the black line is the control, and all the others are different concentrations of the crowders. So we were wondering, it is difficult to interpret experiments at atomistic level of detail, because you get only these kinds of data. But so this is where Natalia came in, and she performed the simulations of this protease in the presence of substrate, and also in the presence of these PEG molecules as well as water molecules. And this system was on the size of roughly, I think, 250,000 atoms. So we ran these simulations for about 500 nanoseconds many times, and I will show you the movie how it looks, if you can see it, if not just the last frame probably. So here are the substrates. Here just are examples of PEG molecules, so you see how big they are. And the concentration here is roughly 18 or 17% of excluded volume. However, bear in mind that cells can be crowded, I think, up to 30 or even 40% with other macromolecules. So we were looking, what are the reasons? And what are the reasons? So for the fact that that the reaction is slowed down. So one of the reasons is that the fusion of the substrate, which is calculated based on the trajectories we generated, is slowed down by about 35% in the presence of Crowders, which you see here in this violet row. And what we found is that it's mainly an excluded volume effect because the substrates do not really interact with polyethylene glycol molecules in terms of hydrogen bonding. Another reason is that the diffusion of the protease is also slowed down. But here, what we found is that PEG molecules, they form many contacts with many contacts many contacts with the protease in comparison with the substrate, and apart from these contacts, we also see a lot of hydrogen bonding. And this was interesting because these Crowders were typically considered as inert molecules that do not really strongly interact with neither with each other nor with the enzyme. So it seems that this slowdown is not only due to specific interactions, but also through some hydrogen bond contacts, which I'm showing here. This is an example of how this PEG molecule, which is this chain, shown like encircling the arginine in the protein complex. We also found, because there is also a question, do PEGs preclude access of the substrate to the active site? But it seems they do not. So by combining these experimental studies and the simulation techniques, we can try to at least answer the question, what is happening at the atomistic level of detail, and be able to help interpret the experiments. Now, the last example, I see I have, I think, 5 minutes. It's a dynamics not of a protein, but RNA, small fragment of an RNA. And this is a fragment of messenger RNA that nature put in non-coding regions in some bacteria to regulate the expression of heat shock proteins. So what happens if you look on the left-hand side and see this red part of the RNA sequence of the mRNA fragment? This red part, in order for the proteins to be expressed, need to be accessible to the ribosome. So the ribosome needs to sit over the small and large subunit, that red part. Now, in principle, at ambient conditions, the secondary structure of messenger RNA precludes binding of the ribosome. And upon heat shock, so upon changing of the temperature, this structure unfolds. That's why it's called RNA thermosensor. And the protein downstream is expressed. And this is the case of, and this is how some bacteria respond to heat shock, because what they do, they produce some proteins that preclude that act as a chaperones and preclude destroying the functional proteins. Another thing is that in this way, some virulence genes are also encoded. However, it's not a typical on-off thermal switch. It's more dimmer-like. So it controls a bit the number of protein that is produced. So it's quite, this RNA unfolding dynamics is quite important, and it's quite subtle. So at some point, these are studies of Philippe and Maciej. What we have done is we looked how this RNA unfolding occurs. And I will show you one movie. We simulate this RNA thermal switch. as a function of temperature at different temperatures, MD simulations. Explicit solvent, but to show the movie, I'm showing you only the RNA structure, and I'm showing you upon the temperature raise how this red part gets exposed to the ribosome. Okay, so this is the unfolding part, but what we need to do is not just to show the movie, but we need to quantify. And perhaps what we wanted to do is quantify how this unfolding occurs depending on the sequence. So here I'm showing examples for the few mutants, how the number of hydrogen bonds from the simulation, from many simulations, because we're taking averages, as a function of temperature changes. And what kind of information we can get out of this is we can map these numbers on the secondary structure of different mutants to see which fragments melt first and which fragments melt last. So to see the unfolding mechanism and try to relate this unfolding mechanism with the protein expression that we have from experiments of others. So this is another example of how even simulations for a smaller system for different kinds of mutants can give us some answers to what might be happening and what kind of sequences should we use to try to achieve protein expression at a lower or higher temperature. Okay, so before I summarize, just if you're interested in other RNA-based switches, there are two lightning talks today about a switch responding to aminoglycoside antibiotics, and another talk, poster talk, is about the mechanism of transport of some molecules to bacterial cells, which also involves a lot of dynamics. And I would like to so the take-home message is that the internal dynamics is crucial for the function of many molecules. Sometimes the temporal scale of this dynamics is quite large. I would like to thank Natalia and Agnieszka for their work on the enzyme HCV enzyme. Philip and Maciej contributed to the RNA thermometer work, and the simulation time was also from ICM sent, and we've also been using SDSC resources. And thank you for your attention.
Thank you very much. We have maybe time for 2 questions. Any questions from the audience? Perhaps I will ask, when you say that solvent, the water was explicitly included, does it mean they sort of at the molecular level or through some dielectric function or continuous model?
In most of the simulations I have shown, it was included as explicit water, mostly explicit solvent. Only in one simulation, this was in a way of a dielectric constant. So typically we include it, but we then we don't show it because then you don't see actually the motions.
So another question I might have.
Is that thermosensor you have shown unique, or is that function also visible at other bacteria? This thermosensor I have shown is turned for you and is unique for Salmonella, but there are also others different sequences in different kinds of bacteria. I'm not sure if this answered.
So I might have another question. You mentioned Anton 2. That's a special purpose supercomputer for molecular dynamics and long trajectories. Could you comment on the advantages of specialized supercomputers like that?
We haven't used Anton. I just mentioned it that they are specialized. So obviously you can simulate long. I mean, they are dedicated. Yes. So the scaling is. I mean, it's much better to use a dedicated computer. Yes. So also in terms of the integration algorithm. Yes, and the stability of the simulations. But we have my group didn't have time on Anton. We didn't apply for this, so we're using mostly the ICM resources.
Okay.
What are the methods of calculating coarse grain? Oh, it's based on the so the potential energy function has to be parameterized for coarse grained models, and typically what we do. We use statistical approaches, so we take information from the protein data bank on structures, and we do the Boltzmann. I mean, we calculate statistics of the pseudo bonds or pseudo angles, and then we use the Boltzmann inversion to obtain the harmonic constants and the potential energy function. So this is the most common approach. So to use statistics from many X-ray or cryo-EM structures.
Thank you very much.